Cooksonia. It appears in the mid-Silurian to early Devonian as one of the first vascular land plants. Vascular means it had internal transport tubes, xylem for water and dissolved minerals, phloem for sugars, allowing upright growth beyond the film of water that non-vascular plants require. The plant formed simple, leafless, dichotomously branching stems that ended in sporangia, capsules that produce spores. Typical individuals were only a few centimeters tall and anchored shallowly. True roots, leaves, and wood were absent. Reproduction relied on wind or water-dispersed spores, a single-celled propagule that does not contain an embryo the way seeds do. Cladoxylopsides, represented by fossil stumps, labeled Eospermatopteris in the Middle Devonian Gilboa forest, introduced tree-like stature without modern wood architecture. Their trunks were built from many discrete vascular strands arranged around a hollow center rather than a single continuous woody cylinder. The crown carried large frond-like branches. Support came from a flared basal root mantle rather than deep taproots. This construction produced trees up to several meters tall that could form stands despite lacking true leaves and seeds. Archaeopteris, a late Devonian progymnosperm, combined fern-like foliage with genuine secondary wood, growth produced by a vascular cambium, and extensive branching roots. It reproduced by spores, not seeds, yet it built the first stratified forest canopies known from the rock record. Wood anatomy in Archaeopteris shows ring-like growth increments and the ability to transport water efficiently to a tall crown, enabling forest formation across multiple continents. Lepidodendron. It was the dominant tree of Carboniferous coal swamps, part of the Lycopside lineage. These plants could reach heights of 30 meters, with trunks more than a meter in diameter. Their bark displayed diamond-shaped scars where leaves once attached, giving them the appearance of scaled skin. Unlike modern woody trees, Lepidodendron produced only a thin outer layer of vascular tissue. The central core of the trunk was soft and weak, so support came mainly from the stiff outer shell. Their growth strategy was rapid. They matured, reproduced, and died within a few decades. Reproduction occurred by spores held in cone-like structures at branch tips. When they died, entire groves collapsed quickly, leaving behind thick mats of vegetation that later formed coal. Sigillaria. This was a close relative of Lepidodendron, with a similar tall, unbranched trunk. Its distinguishing feature was the arrangement of leaf scars in vertical rows running up the stem, giving it a lined, ribbed appearance. The crown carried long, narrow leaves and strobili, spore cones, at the top. Like other Lycopsides, Sigillaria had shallow root systems formed from stigmarian axes that radiated horizontally with attached rootlets. These stabilized swamp soils but did not penetrate deeply, making the trees vulnerable to toppling. Calamites. This species belonged to the Sphenopsid group, related to modern horsetails. They grew up to 20 meters tall, with segmented hollow stems that resembled giant bamboo. The stems branched into whorls of needle-like leaves, while underground rhizomes allowed the plants to spread clonally, creating dense thickets. Growth rings preserved in their stems indicate rapid vertical growth in wet environments. They reproduced with spore cones borne at branch nodes, a pattern similar to living horsetails but on a much larger scale. Tree ferns. Tree ferns were another major component of Carboniferous forests. These plants produce tall, trunk-like stems topped by crowns of large, compound fronds. Unlike Lycopsides, tree ferns generated true fronds with a vascular network, similar in appearance to modern ferns, but much larger, often exceeding 3 meters in length. Their reproduction relied on spores released from clusters called sori on the undersides of fronds. Tree ferns could regenerate fronds after damage, enabling them to persist in disturbed swamp habitats. Glossopteris. Glossopteris was a genus of seed ferns that spread across the southern supercontinent Gondwana during the Permian period, about 299 to 252 million years ago. Its leaves were long and tongue-shaped, with a central midrib and a network of side veins. Fossils of Glossopteris leaves are abundant in Permian rocks from South America, Africa, India, Australia, and Antarctica. The wide distribution of this genus later provided key evidence for continental drift, as similar fossils were found on land masses now separated by oceans. Glossopteris reproduced with seeds and pollen organs attached directly to the leaves. This seed-based strategy allowed it to colonize cooler and drier environments compared to the spore-reliant plants of the Carboniferous. Medullosa. This was another unusual seed fern of the late Carboniferous and Permian. It had hollow stems filled with bundles of vascular tissue rather than solid wood. Its leaves, known as Lepidodendron or Neuropteris depending on species, could be several meters long, spreading outward like a giant umbrella. This morphology made the plant look top-heavy, and the weak, spongy stem tissue likely limited its structural stability. Despite this, Medullosa was successful in swampy habitats, where rapid reproduction and vegetative spread compensated for its fragility. Fossil evidence suggests it produced large seeds encased in fleshy outer layers, an early example of investment in protective seed coats. Cordaites. Cordaites were tall seed plants related to the ancestors of conifers. They grew up to 30 meters tall and bore long strap-like leaves that could exceed 1 meter in length. These leaves had parallel veins and a leathery texture, adaptations for withstanding sunlight and drying conditions. Unlike the shallow-rooted lycopsides of the Carboniferous, Cordaites had deep root systems and woody trunks, enabling them to thrive on uplands as well as in swamps. They reproduced with seeds born in cone-like structures, and their wood anatomy shows features similar to conifers, making them an important transitional group between early seed plants and the gymnosperm forests that later dominated the Mesozoic. Bennettitales. These were a group of extinct seed plants that flourished during the Triassic and Jurassic. They resembled cycads in their overall appearance, with stout trunks and crowns of stiff, pinnate leaves, but they produced highly distinctive reproductive organs. Their cones were arranged in structures that mimicked flowers, complete with whorled bracts and spiraled scales. These flower-like organs were not true flowers, as they lacked petals and enclosed ovaries, but they show convergent evolution toward angiosperm reproductive strategies. Fossil cuticles reveal that Bennettitales had thick, leathery leaves, suggesting adaptation to warm and seasonal climates. Their decline began in the Cretaceous as flowering plants spread, and the entire order disappeared by the end of the period. Cycads. Cycads were another major component of Mesozoic vegetation, and they survive in reduced diversity today. They are slow-growing woody plants with stout trunks and crowns of stiff leaves arranged like rosettes. During the Mesozoic, they were widespread and diverse, often dominating landscapes in tropical and subtropical belts. Many cycads contain toxic compounds such as cycacin, a glycoside that disrupts the nervous system and liver in animals. These chemical defenses made them unpalatable or dangerous to many herbivores. Fossil evidence shows cycads were important food sources for some dinosaurs that may have evolved tolerance or specialized feeding strategies. Conifers. Conifers formed the backbone of Mesozoic forests. Groups such as araucarians, relatives of the modern monkey puzzle tree, grew into massive, straight-trunked canopy trees with whorls of horizontal branches. These plants produced tough, resinous wood and scaled leaves. Their resin acted as both a chemical defense against insects and fungi, and a physical sealant for wounds. Fossilized resin, known as amber, often preserves insects and plant material, providing a detailed record of ancient ecosystems. Other important conifers included podocarps, Charolipediaceans, and early relatives of pines. These lineages diversified into both lowland floodplains and upland slopes, making them ecologically dominant for over 100 million years. Ginkgoes, represented today by the single living species Ginkgo biloba, were far more diverse during the Mesozoic. They are recognized by their unique fan-shaped leaves with radiating veins. Fossils show that Mesozoic ginkgos formed both small trees and large canopy species. They reproduced with ovules borne in pairs on stalks rather than in cones, producing fleshy-coated seeds that resemble fruit but are gymnosperm structures. Ginkgo leaves are found in Jurassic and Cretaceous deposits worldwide, showing that the group was once widespread. Their distinctive form and unusual reproduction make them a classic example of a living fossil, persisting almost unchanged for over 200 million years. Angiosperms. The appearance of angiosperms or flowering plants during the early Cretaceous marked one of the most significant evolutionary transitions in plant history. Unlike gymnosperms, angiosperms enclosed their seeds within ovaries that developed into fruits. Their reproductive organs included flowers with petals and sepals, which both protected developing structures and attracted pollinators. Fossil evidence of early forms such as Archaeopteris from China, dated to about 125 million years ago, shows elongated stems with small flowers and seeds enclosed in carpels. These plants lacked showy petals, but demonstrated the fundamental angiosperm trait of enclosed ovules. Angiosperms diversified rapidly in the mid-Cretaceous. Their success was due to several advantages. First, flowers allowed precise pollination, often involving insects, reducing waste compared to wind-dispersed pollen. Fossilized pollen grains confirm that by 100 million years ago, flowering plants had spread across many environments. Second, the evolution of fruits provided both protection for seeds and mechanisms for dispersal by animals, wind, or water. Some early fruits were fleshy, while others developed hard shells or wings. Third, their leaves typically had broad blades with complex vein networks, enabling efficient photosynthesis and faster growth compared to conifers and cycads. Defensive strategies of angiosperms were diverse. Many lineages developed thorns or spines to deter browsing. Others produced potent chemical defenses in the form of alkaloids, tannins, or latex. These compounds discouraged herbivory by making tissues toxic, bitter, or indigestible. Fossil evidence of leaf damage patterns in the Cretaceous shows that insects and herbivorous dinosaurs quickly adapted, leading to an evolutionary arms race. Some plants increased toughness by producing silicified tissues, while others relied on rapid growth and seasonal shedding to reduce losses. By the late Cretaceous, angiosperms had become dominant in many regions, gradually displacing gymnosperms from lowland and disturbed habitats. They formed forests, open woodlands, and flowering understories, adding structural and ecological complexity to ecosystems. Their coevolution with pollinators, particularly insects such as beetles, flies, and early bees, established long-lasting mutualisms that continue today. Angiosperms also influenced animal diets, providing fruits and seeds that supported new niches for herbivores and seed dispersers. The rapid expansion of angiosperms fundamentally reshaped global vegetation. Gymnosperms, while still present, lost their overwhelming dominance. Flowering plants' capacity for fast reproduction, varied defenses, and flexible growth made them the architects of modern terrestrial ecosystems, a role they assumed before the end of the Cretaceous.